Disease can be the consequence of both infectious and non-infectious processes, and disease is always present in a population. How do health professionals determine if a disease outbreak is caused by an infectious agent? This is a good time to review some of the fundamental concepts of infectious disease, because that's where the clues to determining the cause of an infectious disease outbreak can be found. Recall that the key features of an infectious disease is that there is a microorganism, we call this a pathogen, responsible for the clinical signs of the disease, and that this pathogen can replicate in one individual and be transmitted to another. With this information, the process of determining whether a disease outbreak is due to an infectious agent is straightforward. Let's see how this works. The investigation starts when health professionals notice that there is something different about disease symptoms, or the number of individuals with specific disease symptoms. For example, there could be reports of unusually severe disease, or a disease that is common in some geographical areas could appear at a different time of the year or in a new region. Medical professionals are key to identifying infectious disease outbreaks because they are likely to be the first to recognize that there is something unusual in the symptoms that their patients are experiencing or in the number of patients with those symptoms. In 1999, it was astute doctors in New York City who reported that there was an increase in the number of patients with encephalitis, which set off the public health investigation that identified West Nile virus emergence in the US. A good description of the clinical features of the disease under investigation is needed to determine if an infectious agent is involved. Once the clinical case history is known, medical and public health professionals can identify other individuals with similar symptoms to determine if there is a contact history among individuals, which would suggest an infectious organism, and to obtain samples to search for the infectious organism. Once the disease is recognized and clinically characterized, the next step is to determine if there is an infectious agent present. The process of demonstrating that a transmissible organism can cause disease outbreaks has its roots in an era lacking today's technological advances. In the late 1880s, Dr. Robert Koch proposed that to establish a causative link between a pathogen and a disease, the pathogen should be present in all individuals with a defined set of symptoms, and the same clinical symptoms should occur in healthy individuals who have been inoculated with the organism isolated in pure form from the sick individual. This is the basis for Koch's postulates, which have long guided investigations establishing that an infectious agent causes a disease. Similarity in clinical signs in the donor and recipient of the organism are the key factors here. But Dr. Koch also required that a pure organism be transmitted to assure that only the isolated microbe caused disease. So how are pathogens isolated? Pathogens in samples taken from an infected person can sometimes be induced to grow in the lab given the right cells or nutrients. If culturing is successful, the pathogen can be purified and used to infect other individuals to fulfill Koch's postulates. Having a pathogen in culture also aids the development of diagnostic reagents so that infected individuals can be identified, and of therapeutic reagents. Sometimes microbes won't grow in culture or grow very slowly. Although it's difficult to establish that a microbe is transmissible without having it isolated, today there are 3 alternative approaches to identify pathogens, even if they're difficult to grow in culture. A reliable first approach to detect a microorganism is to actually see it. Typically, this is done using a microscope. Very small organisms like viruses require high-resolution microscopes. Fortunately, many virus families can be identified by shape, so a microscopic image can provide a good lead as to what to include in a potential lineup of suspects during an outbreak investigation. If microscopic data and disease symptoms point to a member of a known pathogen family, it's possible to determine if a sick person was infected with the pathogen by detecting antibodies in the blood. Antibodies are proteins produced by a B cell, part of the adaptive immune system, that bind to short segments of pathogen molecules called epitopes. The specificity of antibodies for pathogen molecules can be exploited both to classify the agent and to confirm that an individual who is sick is infected with that agent. Thus, even without the pathogen in culture, similarity in the antibody profile of two individuals is supportive evidence that they were infected with the same pathogen. A remarkable advance that is transforming outbreak investigation comes from new sequencing technologies. These have dramatically increased our ability to detect very small amounts of pathogen genes. Significantly, these so-called next-generation sequencing approaches don't require any knowledge of specific genetic sequence of the pathogen because all nucleic acid in the sample is sequenced. Next-generation sequencing approaches will likely complement or replace more conventional approaches for disease outbreak investigations in the next few years because of their speed and sensitivity. Today, health professionals have many more tools available to detect an infectious disease outbreak than our predecessors did. The key data needed are evidence that the pathogen can be transmitted from a sick to a healthy individual and produce the same disease, that the microorganism is present in individuals who have common disease symptoms, and the identity of the causative organism.